When the lysate is used to infect the population of recipient cells, most of the cells become infected with normal viruses. However, a small proportion of the population receives transducing particles that inject the DNA they received from a previous host bacteria. While this DNA cannot replicate, it cannot undergo genetic recombination with the DNA of the new host because only a small proportion of the particles in the lysate are of the defective transducing type, and each of these contains only a small fragment of donor DNA. The probability of transducing particles contain particular gene is quite low, and usually only about one cell and 10 to the power of 10, negative 10 to the power of 8, and transducted for a given marker. Phages that form transducing particles can be either temperate or virulent, the main requirements being said that they have a DNA packaging mechanism that permits accidental recognition of host DNA, and that packaging occurs before the host genome is completely degraded. The detection of transduction is most certain when the multiplicity of phage of host is low, so a host cell is infected with only a single phage particle. With multiple infection, the cell will likely be killed by the normal virions in the lysate. Specialized transduction. Generalized transduction that also allows the transfer of DNA from one bacterium to another at a low frequency. However, specialized transduction can allow extremely efficient transfer while also allowing a small region of a bacteria. Chromosome to be replicated independently of the rest. The example we shall use to discuss specialized transduction was the first to be discovered and involves transduction of the galactose genes by the temperate phage Lambda vestigia coli. As we discussed in section 10.9.10, when a cell is lysogenized by lambda, the phage genome becomes integrated into the host DNA at a specific site. The region in which lambda integrates is immediately adjacent to a cluster of host genes that control the enzymes involved in the galactose utilization. The DNA of lambda is inserted into the host DNA at that site. From then on, viral DNA replication is under host control. On induction, for example by ultraviolet radiation, the viral DNA separates from the host DNA by a process that is the reverse of integrase. Ordinary, when the lysogenic cell is induced, the lambda DNA is excised as a unit under rare conditions. However, the phage genome is excised incorrectly. Some of the adjacent bacteria genes, for example the galactose operin, are excised along with phage DNA. At the same time, some phage genes are left behind. One type of altered phage particle called lambda D-gal or DA-gal means defective galactose, is defective because of the phage genes lost and does not make mature phage. However, a helper phage can provide those functions missing in the defective particles. This helper is identical to the original lambda. Thus, the culture lysate obtained contains a few algal particles, lambda particles, in which the large number of normal lambda virions. When a galactose negative bacteria culture is infected at high multiplicity with the slush lysate, and gal plus, transductants are selected. Many are double lysogens, carrying both lambda and lambda D-gal. The bacterium has become a diploid for the gal region. This will have significance in performing complementation tests in bacteria. When such double lysogen is induced, a lysate is produced containing about equal numbers of lambda, and lambda D-gal, such as lysate, can transduce a high efficiency, although only for a restricted group of gal genes. If the phage is to be viable, there is a maximum limit to the amount of phage DNA that can be replaced with the host DNA because sufficient phage DNA must be retained in order to provide the information for production of the phage protein coat and for other phage proteins needed for lysis and lysogenization. However, if the helper phage is used together with the defective phage in a mixed infection, then even less information is needed. In the defective phage for transduction, only the attachment region of the cohesive site for packaging and the replication origin of lambda genome are needed for production of transducing particle provided a helper is used. One important distinction between specialized and generalized transduction is in how the transducing lysate can be formed in In specialized transduction, this must occur by induction of lysogen, whereas the generalized transduction, it can occur either in this way or by infection of non-lysogen by the phage with subsequent phage replication and cell lysis. Although we have discussed specialized transduction only in a lambda-gal system, phage lambda, its specialized transduction in bacteria is similar to the situation we discussed with retrovirus, where certain host genes can be incorporated into the retroviral genome and passed along, often in modified form, to subsequent hosts. These have been detected in retroviruses because the genes involved are oncogenes involved in the development of cancer. Phage conversion. This is a phenomenon analogous in some ways to specialized transduction. When a normal temperate phage, that is a non-defective one, lysogenizes a cell and its DNA is converted to the prophage state, the lysogen is immune to further infection by the same type of phage. This acquisition of immunity can be considered a change in phenotype. In certain cases, other phenotype alterations can be detected in a lysogenized cell, which seem to be unrelated to the phage immunity system. Such a change, which is brought about through lysogenization by a normal template phage, is called phage conversion. Two cases of conversion have been especially well studied. One involves a change in structure of polysaccharide on the cell surface of the Salmonella antigenum on lysogenization with phage. The second involves the conversion of non-toxin producing strains of Corybacterium, the bacterium that causes the disease diphtheria, to toxin-producing pathogenic strains on lysogenization with phage B. In these situations, the information for production of these new materials is apparently an integral part of the phage genome and hence is automatically and exclusively transferred on infection by the phage and lysogenization. Lysogeny probably carries a strong selective value for the host cell because it confers resistance to infection by viruses of the same type. Phage conversion may also be of considerable evolutionary significance because it results in efficient genetic alteration of host cells. Many bacteria isolated from nature are lysogens. It seems reasonable to conclude, therefore, that lysogeny is the normal state of affairs and may often be essential for survival of the host in nature. Plasmids. Before we discuss the third method of genetic transfer, conjugation, we must discuss plasmids. Plasmids are genetic elements that replicate independently of the host chromosome. Unlike viruses, plasmids do not have an extracellular form and exist inside cells simply as a nucleic acid. However, distinguishing between viruses and plasmids can sometimes be difficult, as we have discussed the prophage form of some temperate viruses such as bacteriophage P1 replicates independently of the host chromosome in fashion analogs to plasma replication. We noted earlier that it is possible to differentiate between a plasmid and host chromosome and prokaryotes because plasmids do not carry genes required by the host under all conditions. This can be difficult to prove, and therefore it may sometimes be difficult to distinguish between chromosomes in very large plasmids in prokaryotes. Despite these few difficulties, literally thousands of different types of plasmids are known. Indeed, over 300 different naturally occurring plasmids have been isolated from strains of Escherichia coli alone. In this section, we shall discuss the properties of a few of them. Physical nature of plasmids. Almost all the known plasmids are double-stranded DNA. Most plasmids are circular, but many linear plasmids are also known. Naturally occurring plasmids vary in size from approximately 1 to more than 1,000 kilobase pairs. The typical plasmid is circular double-stranded DNA molecule less than 1/20 the size of the chromosome. Most of the plasmid DNA isolated from cells is in the supercoiled configuration, which is the most compact form within the cell. Isolation of plasmid DNA generally be readily accomplished by making use of the physical properties of supercoiled DNA. Molecules, although chromosomes, are also supercoiled inside the cell. Isolated from a chromosome, DNA almost always leads to breakage of the strands and consequent loss of supercoiling. Separation can then proceed by a variety of techniques, including ultracentrifugation, electrophoresis, agarose gels. Replication of plasmids. The enzymes involved in plasmid replication are normal cell enzymes, so the genes carried by the plasmid itself that control its replication are concerned primarily with the control of the timing of the initiation process and with apportionment of the replicated plasmids between daughter cells. Also, different plasmids are present in cells in the particular number of plasmid molecules per cell. This is called the copy number. Some plasmids are present in the cell in only to 1 to 3 copies, whereas others may be present in over 100 copies. Copy number is controlled by genes on the plasmid and by interactions between the host and the plasmid. Most plasmids are gram-negative bacteria replicate in a manner similar to that already described for the chromosome. This involves initiation of replication at an origin and bidirectional replication around the circle, giving the theta intermediate. However, some plasmids have unidirectional replication. Because of the small size of plasmid DNA relative to the chromosome, the whole replication process occurs very quickly, perhaps in 1 to 10 or less than the total time of the cell division cycle. Most plasmids of gram-positive bacteria replicate a rolling circle mechanism similar to that used by the phage. This mechanism gives rise to single-stranded intermediate, and thus these plasmids are sometimes referred to as the single-stranded DNA plasmids. Most of the linear plasmids now known replicate using a mechanism involving a protein bound to the 5' end and of each strand that is used in priming DNA synthesis. Some individual bacteria cells may also contain several different types of plasmids, for example, Borrelia, Burgdorferi, Lyme pathogen. Contains 17 different circular and linear plasmids. The ability of